Hello cryobiologists, my name is Daniela Impe. I am a PhD candidate in the cryo and stress biology group, and I'm working in the IPK in Gartersleben, Germany. I am interested in wheat pollen, especially after shedding from the anthers, what happens with the metabolites after shedding, whether we can store the pollen, and why viability is lost so fast. What is currently known about wheat pollen? We know that wheat pollen lose viability extremely rapid after they are shed. Biological processes in mature wheat pollen are hardly understood. We know that hybrids have a high potential to increase yields, and that breeding barriers might be overcome by establishing pollen banks. However, we know that preservation protocols have not been established yet. When we want to preserve pollen, we have to consider that storage possibly affects several things. This might be ultrastructure, viability, metabolites, or water content. And overall, changes in those traits might have an effect on the longevity of the pollen during storage and also on the fertility. What do we investigate now? So we want to study in 4 different wheat lines, Perum and Hermann, which are 2 winter wheat lines, and 34399 and 39102, which are 2 land races. And from those 4 wheat lines, we collect pollen from mature anthers, and we store them under 4 different conditions. We have room temperature with low relative humidity and high relative humidity, and cold temperatures with low and high relative humidity. We compare those 4 storage conditions with fresh pollen. What are we doing after storage? We investigate changes in the ultrastructure, changes of viability, the water content, and the metabolites. And first of all, I want to show you a little bit what happens with the ultrastructure when pollen is stored. And here we use a microscopy, light and cryo microscopy, to have an overview of changes that happen, and TEM microscopy, transmission electron microscopy, to study ultrastructural changes of the pollen. And what we see in the light microscope is that after pollen is shed, pollen shrinks very quickly. You can see it also in this pollen here. And this might be a hint that there are damages going on when pollen is stored. And those damages happen within a very short time, only 10 minutes after shedding. And when we have a closer look into the pollen with the ultra-transmission electron microscopy, we see, first of all, in the fresh pollen, they look very round. And the nucleus in the middle picture, it seems that it is smooth, and the compartmentalization of the cell looks normal. We can see different compartments here. However, when we compare the fresh pollen with the dried pollen, for example, pollen that had been stored for 60 minutes in room temperature, we see that the protoplast is a bit disattached from the cell wall. The nucleus seems to be degraded and very unorganized, and the compartmentalization of the cell organelles in the last picture might be dissolved already. So we think that there might be definitely damages after storage. And this, of course, has an influence on the viability. Since we studied it, you might know that there are different methods of the assessment of viability, and we have some traditional methods here, the seed set and in vivo germination, and also some staining methods. However, those 2 methods are either where you have to put a lot of effort to get the results, or you get a lot of false positives. That's why we also studied pollen germination media and impedance flow cytometry, and the comparison of those 4 methods on the viability of pollen we published in 2020 in January. And that's why I would not go further into of these results and want to continue directly with the viability during storage. And we got very interesting results here. When you compare the fresh pollen here in gray with the 4 different storage conditions, you see that under room temperature conditions in beige and light blue here, pollen lose the ability to germinate very quickly. Only after 10-20 minutes of storage, pollen has lost most of its germination. However, under cold conditions here in dark brown and blue, we see that pollen stay viable for longer storage periods. And we saw this effect also with impedance flow cytometry. So here, again, the comparison of fresh pollen with the pollen that has been stored for 60 minutes under the 4 conditions. And here also you see in dark gray, the viability measured with the IFC is lost quickly. What happened now with the water content? So you saw that the pollen shrinks a lot after they have been shed. And this is of course a result of the loss of water content. Here in gray, again, the fresh pollen have a very high water content. However, pollen that has been stored under relative humidity and room temperature conditions have very low water contents. And this reduction in water content also contributes to a loss of viability, what you can see in the second graph. So especially in pollen germination, the lower the water content, the less pollen germinate. And now I want to introduce a little bit about the metabolites and how they may contribute to loss of viability. Again, a little bit about the methods that we used. So we studied, as I said, 4 different wheat lines. We collected the pollen, stored them under the 4 conditions, and then we used GCMS and HPLC analysis to extract metabolites. And from the raw data set, we processed and normalized them and did the statistical analysis. And we used MetaboAnalyst, RStudio, and ClassWiz to, for example, do a multidimensional reduction and compare different levels of metabolites under different conditions. And we found that we have around 40 substances in pollen that we could measure. Among them also we have 4 unidentified substances, and we found carbohydrates where fructose, glucose, and sucrose are the main sugars. We have found amino acids and amino acid derivatives. We found some carboxylic acids, for example, from the TCA cycle. We found some amines, alcohols and polyols, some fatty acids and conjugates, and also some other compounds. And first we saw that when we compare the 4 different wheat lines, that in between the wheat lines, when they are stored, not so many changes happen. This is because the PLSDA analysis is a multidimensional reduction similar to PCA. We saw that the wheat lines overlap a lot. Only between ferrum and 39102, we saw a little bit separation of the wheat lines. However, when we compare storage in the 4 different conditions and what happened to the metabolites, we see that the longer the pollen is stored, the more they are different, the metabolites differ from fresh pollen, which means that there are a lot of changes. In the next slide here, you can see an overview of the important pathways. In green, you have glycolysis, in purple, TCA cycle, and in blue, you see some amino acids. And in the next 3 slides, I will go a little bit into detail of the changes within those pathways. First, we saw changes in the sugars, for example, glucose, sucrose, and fructose, increase during storage, especially in the room temperature with low relative humidity, whereas sugar phosphates, glucose 6-phosphate and fructose 6-phosphate, they decrease during the storage. This might be a hint of, for example, phosphate depletion. Next, we have the TCA cycle, and here we see also an increase of metabolites, mainly of isocitrate, succinate, and fumarate over here. And again, room temperature conditions seem to be more damaging to the pollen, and metabolites increase more in these conditions. This might be also a hint that the pollen needs a lot more energy when they are stored under those conditions. Lastly, we saw also changes in amino acids. Here in the middle, for example, alanine, leucine, and valine. We also saw increases of those amino acids. For proline, on the very right side over here, it is already known that proline is an indicator for abiotic stress. So of course we consider the pollen to be highly stressed during storage. Finally, of course, there are open questions that need to be solved. We consider that the increases of sugar might come from a possible starch degradation. Right now we are studying whether this is really happening by a starch analysis. Further, we want to know whether transcriptional changes contribute to viability loss or to a change of metabolites. We want to know whether proteins are degraded during storage. The pollen wall might be a weak point also, and because the pollen, as you could see, cannot survive short-term storage, we want to work on a long-term preservation protocol using cryopreservation. And here we already studied, made an ultrastructural analysis, and we saw what happened in pollen when they are cryopreserved. So here you have some freshly shed pollen. Let's see what happens when they're in cryo. What you can see first is around the pollen you see ice crystal formation, and then they are darkening and getting very, very black. And this might be a hint that there are ice crystal formation inside the pollen. And when they are thawing, they seem to have changed a little bit also in their structure. And you see there's a lot more to do. And finally, I want to give you some flashbacks from the last cryo conference in San Diego. It was really nice, and I'm very happy that we, this year with all the corona crisis, still have the opportunity to share our results and discuss about them. And there are, of course, many persons to thank. First of all, Manuela Nagel, who is my supervisor, Traut Winkelmann, who is my professor in Hanover. I want to say thanks to Till Ischebeck and Hadi Rolecek, who assisted me in the metabolites analysis. A big thank you also for Michael Melzer, who helped me with the TEM microscopy. A big thanks also to Daniel Ballesteros, who supervised me during my time in Wakehurst, which was also very nice. And of course, there are many more persons that I can't mention now, but thanks again for watching my video, and I'm happy to take your questions during the live course. Thank you.